Thank you. This is Dr. Sylivia on the floor. Please feel at ease. So we're going to go through lipoproteins. Lipoproteins, they are just conjugated proteins together with lipids. When I say that they are conjugated proteins, I'm meaning that it's a protein and on the top of it, we are adding a lipid. It's like a protein being added onto a lipid. So the protein part of a lipoprotein is called an apoprotein or an apolipoprotein. And these protein parts, I'm meaning that the apoprotein, this protein part, it can be either structure or transferred. Structure in that you cannot transfer it, it's within the structure, and it can be transferable. That is, you can remove it from one apoprotein, or you can remove it from one lipoprotein onto another, and that can be maybe after breakdown. So the lipid part also differs depending on the type of the lipoprotein, just like the way the protein part differs. So different lipid components in various, it's different in various components, where we're going to see with further details, you're going to see that the chylomicron has the highest composition of the lipid component with the lowest composition of the protein component. And then we're going to also see that the HDL has the highest protein component but with the lowest lipid component. Let me proceed to the next slide. So now we're going to see the general structure of a lipoprotein. The general structure of a lipoprotein, as in the way you can see it, it is spherical. But in the spherical pattern, we are seeing that it has an outer coat. The outer coat is just, it's a hydrophilic shell. It's a hydrophilic shell consisting of a monolayer of amphipathic phospholipids. This monolayer of amphipathic phospholipids, in that the phospholipids have a hydrophilic head that is facing towards the aqueous environment, and then we are having the hydrophobic tails projecting inside towards to the hydrophobic core. And within that monolayer, we have what we call the unesterified cholesterol, or what we call free cholesterol molecules within. They are embedded just within the phospholipids, and there are those orange dots you can see. So, and still in the monolayer, the outer shell or the outer coat, we have cholesterol is also, I finished that. We also have apolipoproteins, and those ones, the integral ones or the structural ones, which are embedded within, and it is seen by that blue, the blue substance, that substance that you see. So when I go deeper into the lipoprotein, I'm going on to in the inner core, the hydrophobic core. The hydrophobic core just consists of tags and cholesterol esters or esterified cholesterol. So if you want to go deeper into the understanding, it's like as if a lipoprotein is made in that they are getting what we call hydrophilic substance enclosed on what we call a hydrophobic substance inside. So inside we have what is hydrophobic, and outside we have what we call an amphipathic, with the hydrophilic part facing outside. This is the general structure. It's what I've explained in brief. So this is drawable, while this is not drawable. With this, you can draw it. What you see as this is just the integral apolipoprotein. It's the one that traverses inside, and then this, that is a peripheral. It's lying outside, the way you can see. Then afterwards, we also have tags. The way you can see them, they are inside. They are hydrophobic. Then we also have esterified cholesterol. You're seeing it also within the unesterified cholesterol within the monolayer. Let me proceed. So here now we are just going through physical properties of lipoproteins. Under these physical properties, we are comparing density, diameter, lipid component, tags, cholesterol esters, cholesterol itself, the free cholesterol or unesterified, then phospholipid component. And these are some of the physical properties that enable us to identify the different lipoproteins. So as you go peruse through, you can see that a chylomicron has the highest lipid component but lowest. All of this, all you need is to just identify. We're going to come back to this. Let us first go forward. So this is the description just of lipoprotein, and its function is to transport tags. Tags are hydrophobic. They are just enclosed, they are sandwiched inside. So being that they are hydrophobic, they cannot be transported within blood. They have to be encircled by something that is hydrophilic and can be transported within the bloodstream. So lipoproteins majorly transport tags, of which tags are the major sources of energy to the different vital organs of the body. They also transport cholesterol, esterified cholesterol that is within, then together with phospholipids around the body. Then I think we went through this. So now when we dive deeper into the apoproteins, we have 5 major classes of apoproteins, or the protein parts. We have ApoA, ApoB, ApoC, ApoD, and ApoE. And each of the apoproteins, or the apolipoprotein, they have what we call a subclass. Subclass 1, subclass 2, 3, depending on which one. So under A, we have ApoA1 and A2. Then ApoC, ApoC, C2, C3, and others, as we're going to see more. So these are some of the general functions. I think that like the major ones. They are required, the apoproteins are required for structure purposes, like the way you see in a lipoprotein. Apoproteins form what we call the structure, then they are activators of different enzymes. For example, ApoC2 and ApoC1, they activate what we call lipoprotein lipase, while others can act as inhibitors, that is ApoA2 and ApoC3. They inhibit lipoprotein lipase. Then they also act as recognition sites. So these are the general roles of lipoproteins. Enzyme cofactors, as we've gone through. Structure, we've still gone through it. Then still they act as ligands for interaction. They act as ligands for interaction with the different apoprotein receptors in the tissues, for example, ApoB100 and ApoE. Let us try to summarize and be a bit briefer. So, different lipoproteins have different apoproteins that are associated. You're going to see that ApoC1 associates with very low density lipoproteins, high density lipoproteins. ApoC2 associates with chylomicrons and VLDLs. Then HDL function is that lipoprotein lipase. Oh my god. Let us go to the next statement. ApoC3 associates with chylomicrons and VLDLs plus HDLs. Well, the above statement of HDL function activates lipoproteins is that ApoC2 is the one that activates lipoprotein lipase, and then this function that inhibits lipoprotein lipase is caused by ApoC3. So now we are going to see to have types of lipoproteins, and lipoproteins right now they are grouped into 2. That is depending on the size and density, then electrophoretic mobility. So let us go to to size. Lipoproteins, they have the largest size diameter, but they have the lowest density. So they're the ones above, followed by very low density, with second to chylomicrons, then low density, then high density beta. High density has the smallest diameter, but with the highest density, which is contrary to the chylomicrons, with a very large diameter but low density, the lowest in fact. Now we're going to go through the composition of LDLs and HDLs, but these ones are just obvious. So here I'm grouping them depending on, this is still the other physical property we were seeing where we're looking at the protein part and the phospholipid part. So now this is just a combination of density together with the diameter. So you can see very much with the diameter, that is what we call the chylomicron. Sorry, these are remnants, but this is the chylomicron itself with the highest density, followed by its remnants. A remnant of a chylomicron is that chylomicron whereby the tags within it have been removed. So a remnant is just a chylomicron whose tags have been removed for maybe breakdown or storage within the adipocyte, depending. So now, next to the chylomicron, we have a VLDL with a second large diameter, but with the least density, second to the chylomicron, then followed by its remnant, which is the IDL, then followed by LDL, and finally HDL with the smallest diameter, but when it comes to density, oh my G, the highest density. So now here we're going to classify lipoproteins depending on electrophoretic mobility. They're separated here depending on the electrophoretic properties that each of them possess. And we're going to see that they're going to be grouped into alpha, beta, pre-beta, broad beta, specifically in this slide. So, wait, hold on a bit. Yeah, this is it. And right now, when we are using electrophoresis to separate the different chylomicrons, we're going to see that towards the anode we have the HDL, the heaviest one, with the highest density. It is known as the alpha lipoproteins now, seconded by the VLDL as the pre-beta, LDL as the beta. But in between the pre-beta and the beta, we have the IDL, which is the remnant, and in this case it is called the broad beta. Then, lastly, it is the chylomicron above. So here we're going to have the metabolic fate of a VLDL. So we have nascent particles of the VLDL being assembled within the liver. And what are those nascent particles? That is the tags and the cholesterol, the ones that make up the LDL. They are just, first of all, they are assembled within the liver. On top of that, we have its associated apoproteins. Remember, the different lipoproteins, another way to identify them is by considering majorly the different apoproteins that make its structure. And in this case of the VLDL, we have ApoB 100, ApoC, and ApoE. They make up its structure. So those are the nascent particles which are going to be assembled within the liver. And upon their assembling within the liver, they're going to be used to make a VLDL. Then the final bit is going to come in when the ApoC and ApoE from the HDL they are being added onto the VLDL particle that is being formed within the liver. Now they form what we call the final VLDL. So right now here we're going to have just digestion, mobilization, transportation of fats. How does it occur? We're going to see that vertebrates obtain most of their fats that is within the diet. They take it within the diet. They just eat it. They take it orally. Then afterwards, they can use it for tissue respiration, that is within the different vital organs, or then they can mobilize it and then store it within the adipocytes. So they can do it in a series of ways. For example, they can convert the carbohydrates to fats, that is within the excess. Upon your digestion, they convert the excess of it to fats, and maybe they are stored within the adipose tissue. So now we're going to see that what we'll find in the hydrophobic core, the precious one, is the tag, which is the vital part from which you obtain energy for the different organs of the body. We're going to see that the tags provide more than half of the energy that is needed by the different vital organs, that is the liver, the heart, skeletal muscle. So what happens is that intestinal lipase, that is an enzyme, is going to convert the tags into MAGs, those are monoacyl glycerides, and DAGs, diacyl glyceride, into free fatty acids and glycerol. That is upon tissue respiration, that is when they are broken down. Now the products of lipase action, they're going to diffuse into the epithelial cells that are lining the intestinal surface. And guess what we are going through is just the transportation. Now once they diffuse within the epithelial cells that are lining, they are going to now be reconverted back into the tags. Now these tags, they are packaged together with the dietary, cholesterol, and specific apolipoprotein to form the different protein aggregates, the aggregates known as the chyromicrons. Now, the chyromicrons, they transport tags because they have the highest component. I'm going to be brief about this transportation. Now, we're going to see how they group. They're saying that combination of the lipid and the protein produce particles of different densities. For example, from chyromicrons to very low density to high density. That is when the different lipid components combine with the protein to produce different particles. That is when we are considering the physical properties. Now, upon their transportation, the protein moieties of the lipoproteins are recognized by receptors. They are those, remember, among the functions of apoproteins, we saying that they act as recognition sites. Now here they are doing their job, whereby on different cell surfaces that are going to take up those lipoproteins, they are going to recognize the apoproteins by the receptor surfaces they have. Now, for chyromicrons specifically, they are going to move from the intestinal mucosa, they are going to enter into the lymphatic system. From where the lymphatic system, they are going to enter into the blood. The blood now is going to carry them to they're going to undergo different pathways. That is, they're going to be maybe transported to an organ. For this case, they are maybe it's a muscle which is contracting and it needs energy. Then maybe adipose. When it's going to an organ or a muscle, that is when we're going to see the tags being broken down to get energy. Now when it is adipose, that is storage. Now what happens in the muscle in case they're going to break down a tag? The chyromicrons, these chyromicrons, upon reaching the muscle, they have what we call ApoC2 on their structure. Now this ApoC2 is the one that is going to activate an enzyme in the muscle, lipoprotein lipase. Lipoprotein lipase, upon its activation, it is going to break down the chyromicrons, hydrolyzing the tags that it contains to fatty acids and glycerol to obtain energy. That is within the organ or the muscle. Now the remnants of the chyromicrons, those are the chyromicrons depleted of their tags. but they still contain other vital components like the apolipoproteins. They're going to travel within blood to the liver, where they're going to be taken up by endocytosis, which is receptor mediated within the liver. Now when they are taken up, they are going to the liver itself is going to release what we call lysosomal enzymes, and these lysosomal enzymes, they are going to break down the lipoproteins, and the cholesterol that it contains is going to be removed out. Now these are plasma lipoproteins. Hold on a bit. I think this one I finished it. Now here they're just going through